Hello, welcome back to the Computational Genomics Lab. We are on level 6, we've leveled up, and we are going to start to run DESEQ analyses and go through there, find genes we would think are statistically significant, and then eventually in level 7 make figures of them. And that will complete our RNAseq adventure, and we'll find lots of results along the way. So let's get started. Just a quick orientation. We're on the Boulder Rinn Lab GitHub on Masterclass. And we go to lessons, and now we are on 06, differential expression. And so we're going to have 6 small chunks to get through all of our RNA-seq results, finding the genes that are going across time, etc., and going through all the main steps you would need to analyze your data if you had RNA-seq show up. Okay, and we're going to start with 01, run DESEQ, which we're going to do right now. And I am here, oh look, but see, I am left over from where we were last time. If we go to working directory, I am now in 05 RNA-seq, but we've leveled up, so I'm going to go to 06, differential expression, and I am going to make this my working directory by doing set as working directory. Cool. Okay, so now we're in the right place. Let's take 01, run DESEQ2. Again, our markdown file here is going to have a lot more detail. You're going to need these packages. They're pre-organized for you, so if you haven't done that yet, you're going to want to install them. There's a video on how to install these as well. And okay, it looks like we're loaded and ready to go. There's a lot more information here on DESEQ, and I just tried to pick the high-level resources that will explain kind of the basics to you and what we're doing. And so I'm going to quickly load from 05 what we finished doing right here. Boom. Okay, so we have our counts matrix and our DESEQ samples. Now, last time we made the counts matrix filtered to not have zeros, and this is basically that. You can see it is not rounded, so we're going to have to do that again. I'm kind of leaving little steps along the way to repeat so we can kind of get familiar with that, and then we can do this. Load this file is probably better. That was just to show you, be careful what you load. And now we have our counts filtered here, which is what we made previously to pipe into DESEQ. And now, believe it or not, this is how easy it is to run DESEQ. From line 52 to 58 is really all we need. And we need a counts table and a sample sheet, which we have already made. There's our sample sheet, DESEQ samples, telling us what the time point and replicate are, and our counts filtered here, which has our rounded numbers and filtered out for any rows that were 0. DESEQ is going to know if there's zeros, so it's up to you in preference. Check with your lab mates whether they like to filter zeros or not. It's not going to change much. Okay, so now we are going to run this. Again, more detail in our markdown file. But what we're looking at here is we're going to make an object called DDS, which is a DESEQ data structure. And that is going to use the DESEQ functions. This is no longer base R, and why we had to load the DESEQ package, where it's going to say DESEQ dataset from matrix. And this is where it's going to get loaded up and set with its counts file and the sample sheets to sort of coordinate its analysis. And these are the actual names it needs. So count data is going to be our counts filtered, which we made previously and just loaded. Our call data is going to be what are the groups, and that's that DESEQ sample sheet, which will tell us the time point and replicate. And now for design, this is where DESEQ is so powerful, is you can switch this up. We could add, right now we're going to say we're looking for genes that are significant from 0 to 12 hours, 0 to 24, 0 to 48, and 0 to 96. But we could also put in here a plus replicate like this, and it would say, okay, it has to be different in these time points and controlled within the replicates, but for now, just so we can have more genes, we're just going to do time point and find all the genes that are changing across time. So now notice this tilde sign, that means the design is based on this. If you do some Googling, you'll find there's lots of difficult ways, or more complicated ways to make a design in DESEQ. But there we go, our counts filtered, DESEQ samples, and the design we want DESEQ to run it on. So I'm going to run that. Voila, and it converted it to integer mode. It should have sort of done that when we rounded the counts filtered, but DESEQ wants them to be integers, and so it did that for us, and we can see that out here in the console. A little bit more in the console is basically telling you what you just did, and if there's an error, it's going to pop out here. You might be getting it in your RMD, and it might be annoying you, and you can here do chunk output in console, so that means if there's an error, it's going to come out here and not in your R Markdown screen. Okay, so all we have to do to run DESEQ is simply write DESEQ, the function from DESEQ, to run on this DESEQ data structure. And that's really it. The people who developed DESEQ really did a wonderful job to make this simple and very flexible to lots of different statistical models. And believe it or not, we just did differential expression across 4 different time points against 0. In almost no time. It's almost done here. Whoop, there it goes. Okay, cool. So we now have run DESEQ and done our RNAseq. Okay, next function is results names. This will become more and more relevant as we go. And we can see down here, the output of that is intercept, time point 12 versus 0, 24 versus 0, 48 versus 0, 96 versus 0. So any gene that's in all of these results means it's gone up across every single time point, and we're going to do much more analysis on that, but we've done it. We've figured out what genes are changing, and now we got to see what they are. Okay, so another thing to consider when you first run DESEQ on the full data set is to get R-log counts. This is a normalization function that's going to take things with really low values and put less weight on them. This is going to be more for level 7 when we make heat maps and other things, but I just thought it's good to mention it now, and I'll bring it up again later. And to get an R-log count, so we're going to get the same kind of counts matrix, it's just going to be R-logged. So let's take a look at our counts filtered here, and we can see these kind of values, and then those will change to R-log counts, which is an R-log normalization across the values of count. So low count numbers, when you divide by them across the time points, can make inflated values, and so we're going to kind of de-weight those doing this. And so we can run just the rlog function from DESEQ, again in the thing, give it the object, and blind equals true. There's other parameters, and you can simply do this if you want to know what other parameters there are. You can check out the menu, but for now this will work. Okay, this might take a second. And what that's doing is running all the normalization statistics, and when that finishes, which it just did, we're going to use this function assay to go into our rlog counts. Look at this thing. So this is, again, one of these S4 objects, which looks really confusing. You'll eventually get more and more used to these kind of objects, but let's use assay to pull it out as more of a standard kind of table. Okay, so we're going to now make an rlog counts matrix where we previously had a counts matrix. You could call this rlog filtered counts matrix, might be a better actual name for it. But let's do that and assay it to get the results back out. And so now what we can see is we got this object here, which is all these different numbers and negatives and other things. And that's because we just essentially transformed the data into an rlog space. Okay, that's it. We have now run DESEQ, and all those result names all are tables within tables within tables, and that's what's great about R and why it's better than Excel, is that you have all this metadata with data inside the metadata, and we're going to learn how to go in and dig that out and start finding out what happened in our RNAseq. But congratulations, you've now in just a few steps run the RNAseq pipeline to align the counts, and now DESEQ2 to do differential expression. So we can save this just by simply saying write R data structure. This is a little different than the .r data file we were making. RDS is R data structure, and that's the function, and we're going to save rlog counts matrix is a .rds. So anytime you're saving a single matrix type data file, you might want to save it as an RDS, but it can be embedded in your .r data, and it'll pull out the same. Sometimes things can go wrong, so this is a little safer, but you'll probably be fine with the save rdata function instead of write RDS. Okay, so let's look in our results here, and we can see we have, what did we call it? We called it rlog counts mouse docs long, because that's the data we just are, okay, and that was just updated. So we're good to go. Awesome. You're going to run DESEQ, and next we're going to dig in and start analyzing it. All right, we'll see you then.